Hi my dear students, so welcome back to my channel. So welcome back to 2023 science examination preparation video as well. So I think many of you are waiting for the model paper. So ಎಲ್ಲರೂ ಕೂಡ model paper ಗೆ wait ಮಾಡ್ತಿದ್ರಿ ಅಂತ ಅನ್ಕೋತೀನಿ. So ಕರ್ನಾಟಕ ಬೋರ್ಡ್ ಇಂದ ಅಂದ್ರೆ ಕರ್ನಾಟಕ ಎಜುಕೇಶನಲ್ ಎಕ್ಸಾಮಿನೇಷನ್ ಬೋರ್ಡ್ ಇಂದ ಅಫಿಷಿಯಲ್ ಆಗಿ ಇವತ್ತು ನಮ್ಮ ವೆಬ್ಸೈಟ್ ನಲ್ಲಿ ಸೈನ್ಸ್ ಮಾಡೆಲ್ ಪೇಪರ್ಸ್ ನ ಅಂಡ್ not only science ಎಲ್ಲ ಸಬ್ಜೆಕ್ಟ್ ನ ಮಾಡೆಲ್ ಪೇಪರ್ಸ್ ನ ರಿಲೀಸ್ ಮಾಡಿದ್ದಾರೆ. they have released all the subjects model papers. So as I am handling science and I am training for the science examination preparation, ನಿಮ್ಮ ಪಬ್ಲಿಕ್ ಎಕ್ಸಾಮಿನೇಷನ್ ನ ಒಂದು ಸೈನ್ಸ್ ಎಕ್ಸಾಮಿನೇಷನ್ ಪ್ರಿಪರೇಷನ್ ಗೆ ನಾನು ನಿಮಗೆ ಟ್ರೈನ್ ಮಾಡ್ತಿರೋದ್ರಿಂದ so in this video I am going to discuss about the key answers of 2023 science model paper. So before discussing the key answers of the model paper, I would like to discuss about the blueprint. So blueprint ಬಗ್ಗೆ ರಫ್ ಆಗಿ ಹೇಳ್ತೀನಿ ನೋಡಿ. ಸೊ ಈ blueprint ನಾನು ನಿಮ್ಮ ಜೊತೆ ಈಗ ಏನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ, ಸೊ ದಟ್ ವಿಲ್ ಡೆಫಿನೆಟ್ಲಿ ಹೆಲ್ಪ್ ಯು. ಓಕೆ, ಸೊ ಈಗ ಬ್ಲೂಪ್ರಿಂಟ್ ನ ನೋಡೋದಾದ್ರೆ ನಮಗೆ ಕೆಮಿಸ್ಟ್ರಿನಲ್ಲಿ ಟೋಟಲಿ ಕೆಮಿಸ್ಟ್ರಿಗೆ 25 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಓಕೆ, ಸೊ 25 ಮಾರ್ಕ್ಸ್ ಇಸ್ ಅಲಾಟೆಡ್ ಫಾರ್ ಕೆಮಿಸ್ಟ್ರಿ ಟೋಟಲಿ. ಸೊ ನೀವು ಇದನ್ನ ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಎವ್ರಿಬಡಿ ಡೆಫಿನೆಟ್ಲಿ ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಹೆಲ್ಪ್ ಯು. ಓಕೆ, so this will definitely going to help you. So chemical reactions and equations ಗೆ totally 7 marks ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. ಸೊ ಇದನ್ನ ನಾನು ನಿಮ್ಮ ಜೊತೆ ಟೆಲಿಗ್ರಾಮ್ ಗ್ರೂಪ್ ನಲ್ಲೂ ಕೂಡ ಶೇರ್ ಮಾಡ್ತೀನಿ. ಕೆಮಿಕಲ್ ರಿಯಾಕ್ಷನ್ಸ್ ಅಂಡ್ ಈಕ್ವೇಷನ್ಸ್ ಗೆ 7 ಮಾರ್ಕ್ಸ್ ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. ಆಸಿಡ್ಸ್ ಬೇಸಸ್ ಅಂಡ್ ಸಾಲ್ಟ್ಸ್ ಈ ಲೆಸನ್ ಗೆ 5 ಮಾರ್ಕ್ಸ್ ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಆಸಿಡ್ಸ್ ಬೇಸಸ್ ಅಂಡ್ ಸಾಲ್ಟ್ಸ್ ಈ ಲೆಸನ್ ಗೆ 5 ಮಾರ್ಕ್ಸ್ ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. ಸೊ ಮೂವಿಂಗ್ ಟು ನೆಕ್ಸ್ಟ್ ಲೆಸನ್, metals and non-metals. So sorry, acids and bases salts ಗೆ 3 marks ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. metals and non-metals ಗೆ 5 marks ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. Moving to carbon and its compounds, carbon and its compounds ಈ ಲೆಸನ್ ಗೆ 6 marks ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. and periodic classification ಗೆ 4 marks ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. So totally for chemistry it is 25 marks. So 25 marks ಅನ್ನು chemistry ಗೆ allot ಮಾಡಿದ್ದಾರೆ. So out of which chemical reactions and equation lesson ಗೆ 7 mark, acids bases ಗೆ 3 mark, metals and non-metals 5 mark, carbon and its compounds 6 marks, periodic classification of elements 4 marks. So totally 25 marks for chemistry. So chemistry ಗೆ 25 marks ಅನ್ನು allot ಮಾಡಿದ್ದಾರೆ. ಓಕೆ, ಸೊ ಇದು ನಿಮ್ಗೆ ಹೆಲ್ಪ್ ಆಗುತ್ತೆ, ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ಸೊ ಯಾವ ಲೆಸನ್ ಅನ್ನ ನೀವು ಫಸ್ಟ್ ಕವರ್ ಮಾಡ್ಬೋದು ಅಂತ ಡೆಫಿನೆಟ್ಲಿ ನಿಮ್ಗೆ ಇದು ಯೂಸ್ ಆಗುತ್ತೆ. ಸೊ ಮೂವಿಂಗ್ ಟು ನೆಕ್ಸ್ಟ್, ಈಗ ನಾವು ಬಯಾಲಜಿ ಬಗ್ಗೆ ಮಾತಾಡೋಣ. ಸೊ ಬಯಾಲಜಿ ಬಗ್ಗೆ ಮಾತಾಡೋದಾದ್ರೆ, ಸೊ ಬಯಾಲಜಿನಲ್ಲಿ ಫಸ್ಟ್ ಲೆಸನ್ ಲೈಫ್ ಪ್ರಾಸೆಸ್. ಸೊ ಲೈಫ್ ಪ್ರಾಸೆಸ್ಗೆ ಟೋಟಲಿ 7 ಮಾರ್ಕ್ಸ್ ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ಲೈಫ್ ಪ್ರಾಸೆಸ್ಗೆ 7 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ನೆಕ್ಸ್ಟ್ ಕಂಟ್ರೋಲ್ ಅಂಡ್ ಕೋಆರ್ಡಿನೇಷನ್ ಈ ಲೆಸನ್ಗೆ 3 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಹೌ ಡು ಆರ್ಗಾನಿಸಮ್ಸ್ ರೀಪ್ರೊಡ್ಯೂಸ್ ಈ ಲೆಸನ್ಗೆ 7 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಹೆರಿಡಿಟಿ ಅಂಡ್ ಎವಲ್ಯೂಷನ್ ಈ ಲೆಸನ್ಗೆ 5 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಅವರ್ ಎನ್ವಿರಾನ್ಮೆಂಟ್ ಈ ಲೆಸನ್ಗೆ 2 ಮಾರ್ಕ್ಸ್. ಸಸ್ಟೈನಬಲ್ ಮ್ಯಾನೇಜ್ಮೆಂಟ್ ಆಫ್ ನ್ಯಾಚುರಲ್ ರಿಸೋರ್ಸಸ್ ಈ ಒಂದು ಲೆಸನ್ಗೆ 3 ಮಾರ್ಕ್ಸ್. ಸೊ ಟೋಟಲಿ ಬಯಾಲಜಿಗೆ ನಿಮಗೆ ಅಲಾಟ್ ಆಗಿರುವಂಥ ಮಾರ್ಕ್ಸ್ 27. ಸೊ ಟೋಟಲಿ 27 ಮಾರ್ಕ್ಸ್ನ ನಿಮ್ಮ ಬಯಾಲಜಿಗೆ ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ದಿಸ್ ಇಸ್ ಆಲ್ ಅಬೌಟ್ ಬಯಾಲಜಿ. ಸೊ ನಾವು ಲೆಟ್ಸ್ ಗೋ ಫಾರ್ ಫಿಸಿಕ್ಸ್. ಸೊ ನೀವು ಮಾರ್ಕ್ಸನ್ನು ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಳ್ಳೋದ್ರಿಂದ ಸೊ ಯಾವ ಲೆಸನ್ ಕಂಪ್ಲೀಟ್ ಆಗಿದೆ, ಸೊ ಎಷ್ಟು ಮಾರ್ಕ್ಸ್ ಆಲ್ರೆಡಿ ನೀವು ಪ್ರಿಪೇರ್ ಆಗಿದ್ದೀರಾ ಅಂತ ಒಂದು ಕಾನ್ಫಿಡೆನ್ಸ್ ಬರುತ್ತೆ. ಮೂವಿಂಗ್ ಟು ನೆಕ್ಸ್ಟ್, ದಟ್ ಇಸ್ ಫಿಸಿಕ್ಸ್. ವೆನ್ ವಿ ಟಾಕ್ ಅಬೌಟ್ ಫಿಸಿಕ್ಸ್, ಎಲೆಕ್ಟ್ರಿಸಿಟಿ ಲೆಸನ್ಗೆ 7 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಸೊ ಮೂವಿಂಗ್ ಟು ನೆಕ್ಸ್ಟ್, ಮ್ಯಾಗ್ನೆಟಿಕ್ ಎಫೆಕ್ಟ್ಸ್ ಆಫ್ ಎಲೆಕ್ಟ್ರಿಕ್ ಕರೆಂಟ್. ಸೊ ಈ ಲೆಸನ್ಗೆ 6 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಲೈಟ್ ರಿಫ್ಲೆಕ್ಷನ್ ಅಂಡ್ ರಿಫ್ರಾಕ್ಷನ್ ಈ ಲೆಸನ್ಗೆ 7 ಮಾರ್ಕ್ಸ್ ಅನ್ನು ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಅಂಡ್ ದೆನ್ ಮೂವಿಂಗ್ ಟು ಹ್ಯೂಮನ್ ಐ ಅಂಡ್ ಅ ಕಲರ್ಫುಲ್ ವರ್ಲ್ಡ್. ಸೊ ಫಾರ್ ದಿಸ್ ಲೆಸನ್ 5 ಮಾರ್ಕ್ಸ್. Last, sources of energy 3 marks. So this is based on the 2023 model paper. To 2023 model paper ಅಲ್ಲಿ ಈ ರೀತಿ ಮಾರ್ಕ್ಸ್ ಅನ್ನ ಅಲಾಟ್ ಮಾಡಿದ್ದಾರೆ. ಓಕೆ, ಸೊ ಎಸ್, ಸೊ ಮ್ಯಾಮ್ whether this model paper is released by government ಅಂತ you're adding the comments here. So definitely this is been released by our Karnataka Educational Examination Board. So this is an officially released model paper. And according to officially released model paper, I have said the blueprint, that means the marks that you can expect from different lessons. Okay, So now let's move on for the key answers of model paper. So am I audible? Yes. So let us now discuss about the key answers of model paper. So when I discuss about the key answers of model paper, let me start with the chemistry part, followed by biology and then physics. Okay. So first I am going to teach you the answer, or I am going to discuss the answer from the chemistry part. Okay. So question paper is quite easy only, the model paper, but the same type of questions you won't expect. Okay. Same type of questions you won't expect in the examination. So this model paper will definitely help you to understand that what type of questions may come in the examination. Okay. Let us look at the first question. So the first question is, yes, the first question is the chemical equation that represents neutralization reaction among the following is. Okay. So there are 4 options. Already I have mentioned the answer here. See, first of all, neutralization reaction means we always have to look for acid base reaction. See, acid combines with base to form salt and water. Acid combines with base to form salt and water is neutralization reaction. So if you look at the 4 options given here, the acid which they have given in the option A is hydrochloric acid, base they have given is sodium hydroxide. When they both combine, they form sodium chloride and water. So remaining option anna nodidre you cannot see any acid and base. See, barium chloride annodu salt agide. So this option cannot be taken. Then manganese dioxide and hydrochloric acid kottidare. So this is also not a base. Silver nitrate hydrochloric acid kottidare. This is also not a base. So the right answer is the first one, sodium hydroxide combined with HCl to form sodium chloride and water. This is a equation which is representing the neutralization reaction. Okay. Moving to next question. The next question is, properties of elements are the periodic functions of their atomic number. See, you have totally 4 laws in the periodic classification of elements. So among the 4 laws, this is the 4th one. Okay, this is the modern periodic law. Okay, so this modern periodic law says properties of elements are the periodic functions of their atomic number. So who proposed this law anta kelidare? So this law has been proposed by Henry Moseley. Mendeleev has proposed Mendeleev periodic law, which we talk about atomic mass. Newlands says that every 8th element has the property similar to that of first one. Dobereiner law is also the different. Okay. So this atomic number is being proposed by the Henry Moseley. Moving to 16th question, the substances that converts the red litmus paper into blue color anta kelta idare. So if it has to convert from red to blue agbekandre, we have to search for the base. So options ಏನು ಕೊಟ್ಟಿದ್ದಾರೆ, sodium hydroxide, potassium hydroxide KOH, distilled water, dilute solution of HCl, concentrated solution of nitric acid anta ಕೊಟ್ಟಿದ್ದಾರೆ. So potassium hydroxide is a base because we have OH minus ions in this. OH ಅನ್ನೋದಿದೆ. So here the option A is the right answer. So this is all about the multiple choice question. So now let us look at the 1 mark question. So 1 mark question ali first question, mention the number of single bonds and also the double bonds present in the structure of C2H5COOH anta kottidare. So this is actually ethanoic acid. See, when I write C2H5, CH5 and COOH, C double bond O and OH. So what are they asking? They are asking how many single bonds are there and how many double bonds are there. Yes to single bond ide, yes to double bond ide anta kelta idare. See, we can observe here, there are totally, see, 1, 2, 3, 4, 5 and 6 and 7 and 8. So these are the places where hydrogen are getting bond with the carbon. So there are totally 8 single bonds, and you can observe there is only 1 double bond. So the right answer is there are 8 single bonds and 1 double bond is present. Okay, yes. So this is a very simple thing. So this kind of questions you can expect in the examination with the different formulas. Okay. So the 18th question they are asking here is, write the electron dot structure of methane. So you have to write the electron dot structure of methane. See, what is methane's formula? The formula of methane is, okay, the formula of methane is CH4. When we write the structural formula, methane's, okay, let me take out. When I write the structural formula of methane, so 1 carbon is surrounded by 4 hydrogen. So this is how we write the structural formula for the methane. But when we write electron dot structure formula, nodi carbon barkolbeku, carbon giruvanta valence electrons anna barkolbeku, 4 valence electrons, and followed by hydrogens. You have to write every hydrogen atom has 1 valence electron. So let us now denote this with the star mark. Okay, so this is how we are going to write the electron dot structure. So you also have to show whether it is obtained whether it has obtained the stability or not. So to show that, you are going to put the circle na haktidira nodi. This carbon has 8 electrons surrounded by it. So by having 8 electrons, now it has obtained the octet configuration. So hydrogen can have only 2 electrons on its body. Hydrogen can have, so hydrogen can have 2 electrons on its body. So this is how we are writing the electron dot structure for the methane. Okay, I hope this will help you definitely. So I will look at your comments afterwards, children. So I will also tell you, so that from which all the lessons they have given different questions like 2 mark question, 3 marks questions and all, I will discuss with you. Okay. So now let us go for the next question. So the next question is the question number 19. Draw the diagram of the apparatus to show the electrolysis of water anta kelidare. So this diagram you have to write. So you have to learn all the diagrams from chemistry: electrolysis of water, action of steam on metal, and reaction of zinc granules with dilute sulfuric acid, electrolytic refining of copper. Any questions they can ask in the examination. So in this exam they have asked electrolysis of water, and particular agi yavude parts na mention madake kelilla illi. So you need to label all the parts here. So now let us go for 2 marks question. So 2 mark questions ali first question is, see, calcium oxide react with water to form the slaked lime. What type of calcium reaction, what is the chemical reaction is, what type of chemical reaction is this anta keltidare. See, calcium oxide, I have mentioned already your answer, calcium oxide is combining with water. When calcium is combining with water, it forms calcium hydroxide. So what you can observe here, this is a type of reaction where 2 or more reactants is combining to form one single product. So what kind of reaction is this? This is an example for combination reaction. So they are also asking you to write the balanced chemical equation for this reaction. So this is how we are writing the balanced chemical equation. So as I have mentioned here, calcium oxide is combining with water to form the calcium hydroxide. Already in my previous video also I told you that you always have to remember the formula of quicklime, that is the calcium oxide. You have to remember. So let me write here. The formula of quicklime is calcium oxide, formula of slaked lime is calcium hydroxide, formula of limestone is calcium carbonate. These 3 formulas you have to remember, because you may get confused sometimes if you don't read which is the formula of slaked lime, which is the formula of limestone, what is the formula of marble. These all things they will ask in the examination. So it is very important to remember. So they also have given another question here. What is the chemical name of bleaching powder? Write any 2 uses of it anta kottidare. See, the chemical name of bleaching powder. What is the chemical name of bleaching powder? The chemical name of bleaching powder here is calcium oxychloride. What is the chemical name of bleaching powder? The chemical name of bleaching powder is calcium oxychloride. It is very important. So the formula is CaOCl2. Okay, and they have also asked you to write the 2 uses of bleaching powder. The 2 uses of bleaching powder is, one thing is it is used in the paper industries to bleach the wood pulp, and it is also used in the textile industries to bleach the cotton. So these things you have to remember. 2 points I have mentioned here. So you have still more points in the textbook. So these are very catchy always. Okay. So it is used to bleach the wood pulp in which industry? Paper industry, and it is used to bleach the cotton in the textile industries. So these 2 are the very important things you have to remember. Okay. So moving to next question. So the next question is, okay, I think you are able to understand all the answers because most of the questions are very, very easy. So the next question is, what is concentrated acid? Name the acid present in the stinging hair of nettle leaves, they are asking. So usually when we talk about acids, bases and salts lesson, you must know what is meant by acid, what is strong acid, what is weak acid. See, strong acid is nothing but concentrated acid. So what is concentrated acid? See, it is a type of acid where we can see more amount of H plus ions in it. Okay, it is a purest form of acid. So why it is pure? Because it is more amount of H plus ion concentration in it. Okay, and moving to next, name the acid present in the stinging hair of nettle leaves. It is none other than methanoic acid. So methanoic acid is present in the stinging hair of nettle leaves. This methanoic acid is also called as formic acid. Okay, either you can write methanoic acid or you can write formic acid. So this is very important. The next question number 22 is, they are asking from the lesson metals and non-metals. So they are asking, give reasons. Ionic compounds have high melting point and high boiling point. Yes, see why they have high melting point and high boiling point? It is because ionic compounds have strong electrostatic force of attraction between the oppositely charged ions. See, first of all you must know how ionic compounds are formed. They are formed by transfer of electrons. There will be a cation and anion. When cation and anion, when they get transferred from one another, what happen? Ionic compounds are formed. So and these have strong electrostatic force of attraction between them. Okay, so they cannot be melted so easily. That is why they have high boiling point and high melting point. Very easy concept. And one more thing is, ionic compounds in solid state do not conduct electricity. Definitely, see, in the solid state, let us take an example of salt. See, salt is our NaCl. Okay, this is an example for ionic compound. So when we pass electricity to salt, what happens? Salt does not pass the electricity because it is in solid state. But when we add water to this, what happen? When I add water to this, salt start to melt. That means it will start to melt. That is, in its molten state it will conduct electricity. Okay, but in its solid state it does not conduct electricity. Why? Because see, ions are held together by a strong electrostatic force and they are not free to move within the solid state. When they are in solid, ions cannot move. Okay, they have strong electrostatic force of attraction. That is the reason they do not conduct electricity. Ionic compounds do not conduct electricity. Okay, so it is very important to know all these things. So let's go for the next question. So the question number 23, draw the diagram of arrangement of apertures, okay, to show the action of stream on metal. Label metal piece and delivery tube. So you want to label this part, let me take out this, okay. So you want to label the metal piece which is given here. So this part you have to label, and also you have to label the delivery tube. Usually they ask this only, okay. So but you must know all the parts of it. So let's go for the next question. The next question is question number 24. The electronic configuration of four elements are given. Okay, so there I have given 4 elements: sodium, sulfur, aluminium and potassium. Okay, write the elements, these elements in the increasing order of their electropositivity they have given. You must know first of all what is electropositivity, children. Electropositivity is nothing but tendency to lose electron. What is nothing but electropositivity? Electropositivity is nothing but tendency to lose electron. It is tendency to lose electron. What happens to electropositivity across the period? When we go from left to right, okay, electropositivity decreases. Why? Because the loss of electron decreases. Okay, but when we go from top to bottom, what happens? The tendency to lose electron decreases because in the outermost shell there are less number of electrons, so it is easy to lose the electrons for the elements. This concept you have to know first. So when we look at these 4 elements, you can see sodium and potassium, okay, they both have only 1 electron in its outermost orbit. So which element you think which is more positive? Okay, see sodium, when we compare sodium, aluminium, sulfur, when we compare sodium and aluminium, first of all, see potassium is more electropositive compared to sodium. Potassium is more electropositive compared to sodium. Okay, first these two, among these two you have to decide which is more electropositive, you have to decide whether sodium is more electropositive or potassium is more electropositive, because see, in both the cases there is one electron in its outermost orbit, okay, but sodium has 3 shells, potassium has 4 shells. So when I compare these two elements, so potassium is more electropositive compared to sodium. So if you decide among these two, other two elements are very easy. See, sulfur and aluminium they have given. Sulfur has 6 electrons. It is very difficult to lose 6 electrons. But for aluminium, it is easy to lose 3 electrons. So when we arrange according to the increasing order, okay, potassium is more electropositive than sodium. Sodium is more electropositive than aluminium. Aluminium is more electropositive than sulfur. Okay. So you have to write this, and you have to also write the reason. What is the reason? See, electropositivity is nothing but, first of all, tendency to lose electron. So across the period, what happened? The electropositivity decreases. But down the group, what happened? The electropositivity increases. That statement you have to compulsorily write. Okay. Yes. So most of you are asking whether this paper will come for examination. Definitely this paper won't be, the same paper will not be given in the examination. So this is an idea that how your final examination will be. Okay. Moving to next question. Silver chloride exposed to sunlight turns grey color. Why they are asking? See, silver chloride. What is the formula of silver chloride? AgCl2. When this silver chloride is exposed to sunlight, when it is exposed to sunlight, what happen? So it will decompose to form silver and chlorine. It will decompose to form silver and chlorine. Okay. So write the balanced chemical equation for this reaction they are asking. See, I have already mentioned here. Okay. First one is, it is decomposing to form silver and chlorine, and this is the equation which you have to represent. And they are also asking what type of chemical equation is this. This is an example for photolytic decomposition reaction. Okay, because sunlight is involved in this decomposition reaction. There are totally 3 types of decomposition. You must learn all the 3 types, my dear students. The next question is, okay, this is a frequently asked question, very easy one. Okay, why does the color of copper sulfate solution changes when an iron nail is dipped in it? See, when we take copper sulfate solution in a beaker, and when we add iron nail to this, what happens? When I add iron nail to this, see, iron is more reactive than copper. So what does iron do? Iron will displace the copper from copper sulfate solution. Very simple. As a result of it, what happens? Copper gets settled on the iron nail. So I have also mentioned the answer here. You can observe, iron being more reactive displaces copper from the copper sulfate solution. So hence what happened? Copper sulfate solution will turn to green color. Okay, and this is the equation. Fe plus CuSO4 gives rise to FeSO4, and the copper get settled on the iron nail. Okay, so very easy question answers these are. So we also have to look at the state preparatory examination question paper for few more ideas related to your examination. Okay, yes. The next question is, the conversion of ethanol to ethanoic acid is an oxidation reaction. Why? See, let us take ethanol. Ethanol formula is C2H5OH. So when I take ethanol and convert into ethanoic acid, that is CH3COOH, so I say this as an oxidation reaction. Why anta kelta idare? It is because here we are using oxidizing agent, that is potassium permanganate, or we can use potassium dichromate, that is K2Cr2O7. Okay, so these are first of all oxidizing agent. They are adding oxygen to ethanol. As ethanol is added by oxygen, it is called as oxidizing agent. Very easy. Next question, what is structural isomer? See, first of all, isomers means they are the compounds which have same molecular formula but different structural formula. And write the structural isomers of butane. See, when I write the structural isomer for butane, this structure you have to write. This is called as isobutane. Very simple, my dear students. See, iso is nothing but a branched chain. What is nothing but iso? Iso is nothing but a branched chain. Okay. Always you have to write isobutane in this way. So this is all about chemistry. Chemistry part is very easy. So you no need to think about most of the questions. They are direct and most of the answers are very easy. Yes. So now let us look at the biology part. So the first question of biology is, see, they have given AIDS is caused by virus. AIDS is caused by virus. So what is caused by what? See, most of the students have tendency to write that if one is caused by virus, another will be caused by bacteria. But it is a wrong assumption. See, both AIDS and warts, they both are caused by the virus only. Okay. But gonorrhea and syphilis, they are caused by bacteria. Okay. Remember, the right answer is virus. The warts are caused by virus. Moving to 28th question. See, algae, small insects, small fish, large fish, human. See, they have given so many organisms here. Arrangement of trophic levels in the food chain are based on what? See, if you look at this food chain, see, there are 4 options. I have already answered here, but you should know why I have answered this. Okay. See, if you look at this food chain, see, these food chain are, first of all, they are in increasing order. Okay. They are in increasing order. If you look at this food chain, see, the options are increasing order of energy availability, increasing order of both energy availability and storage of harmful chemicals, increasing order of the harmful chemicals, okay, and also they have given the 4th option they have given is, so let me go for 4th option. Yes, the 4th option is decreasing order of both energy and availability and storage of animals. Sorry, storage of harmful substances. See, if you look at this, as we move progressively from one trophic level to another trophic level, accumulation of toxic substances increases and the amount of energy transferred from one trophic level to another trophic level decreases. Okay, remember. So, energy decreases, okay, but what happens here? Energy decreases, but toxic substances, accumulation of toxic substances increases. First they are telling energy availability increases. So this is wrong. Energy does not increase. Energy order of, increasing order of both energy. Just now I told energy does not increase. So this option will also be invalid. See, increasing order of storage of harmful chemicals. Yes, when we go like this in a food chain, what happened? The amount of toxic substances get accumulated in more amount. So right answer is option C. Decreasing order of energy availability. Yes, of course, energy will decrease, but harmful substances does not decrease. Okay. So that is why the option C is right answer. So let's go for next question. The next question is now the 1 mark questions from the biology. So draw the diagram showing the structure of closed stomata. See, open stomata, closed stomata, both you learn. So here they have asked just for 1 mark. Okay. So 1 mark we have to write the closed stomata. Next, what is the function of ozone layer? See, ozone layer, it protects the earth from the harmful UV radiations, that is UV rays, which is coming from the sun. So for 1 mark, this much answer is enough. Okay. So next question is from sustainable management of natural resources. Rejuvenating the ancient water harvesting system is being encouraged. Why? Yes, of course, it is because, see, we can recharge the groundwater by cleaning the ponds and lakes, and rainwater harvesting can also be done. Okay. Dredging of dried lakes also be followed. Okay. And we also have to create the awareness campaign for water rejuvenation. See, first of all, they are asking why we have to do, because first of all the answer we can choose this point. Recharge of groundwater by cleaning the ponds and lakes. We can recharge the water, the groundwater. Okay. So if we rejuvenate, if we start to follow the ancient water harvesting system, we can recharge the groundwater level. Okay. This will also help in rainwater harvesting. Okay. These 2 points are enough for 1 mark. Moving to 32nd question. What needs of local people are fulfilled by the forest? See, you might have learnt about the concept called stakeholders. Okay. See, stakeholders are the people, the one who are getting benefit from the forest and benefited to the forest. Here local people, they are talking about the local people. See, local people, they get large quantities of firewood from the forest, they get timber from the forest, okay, and the bamboo is also used for their the shelter purposes. They will collect the bamboos for making the baskets to make the huts, okay, and they also implement the agriculture, okay, and they also get the food by hunting the animals. Okay. So lot of things are there. You can write any 2 or 3 points. Okay. So what is the importance that why the local people are staying in the forest? Because they are getting benefit from the forest. Okay, they get fruits from the forest, they get nuts from the forest, they get medicines from the forest. So due to these reasons, what happened? The people are living in the forest. Okay, once let me read out the answer for you, this question's answer. Okay, the answer is like this. The local people, they get large quantities of firewood, small timber and thatch. Bamboo is used to make the slats for huts and baskets for collecting and storing the food materials. Implements for agriculture, fishing and hunting are largely made of wood. Forest provides sites for fishing and hunting. Okay, minimum 4 to 5 points you write, children, because this is asked for 2 marks. As they have asked for 2 marks, minimum 4 to 5 points you write, that is more than enough. Okay, so this is all about the question number 32. Let's now go for the next question. The next question is question number 33. See, they have given these two pictures. The one picture is representing the Spirogyra and respecting the planaria. If you look at these two pictures, the question is the reproduction methods expressed in both of the above figures are similar or different from each other. They are asking. See, they both are following the asexual mode of reproduction only. So they are similar. Okay, but you have to write a justification for your answer. See, they are following the asexual mode of reproduction, but here in the Spirogyra, what happened here is the organism upon maturation they split into pieces. Spirogyra, upon maturation, it will split into pieces. Each piece of Spirogyra will develop into a new individual organism. Okay, it is not an accidental process. But in case of planaria reproduce the new organism through the accidental process. Here what happens, in case of planaria, organism due to accidental process, they become into pieces, and each piece of this will develop into a new individual organism. Okay, that is the difference. Spirogyra, upon maturation it will split into pieces. Planaria, by accidental process it will split into pieces. But in both the cases, organism upon splitting they will develop into an individual organism. Okay, and both are asexual mode of reproduction. They are following the asexual mode of reproduction. Next is, how is vegetative propagation in the plants useful to the field of agriculture? Definitely, see, vegetative propagation such as grafting, budding, okay, there are so many other types are there, okay, layering, they all help in growing the new plants from the vegetative parts. Okay, so it is an asexual method of reproduction. Here, most of the vegetative propagation is asexual mode of reproduction, because here agriculturalists, by using the parts of plant, they can grow the new plants. Okay, they can grow the new plants by using the parts of the existing plants. So this is definitely going to be helpful for the agricultural people. See, we can also call them as agriculturalists. The next question is the consistency of, see, related to DNA, one question will be, you can expect, consistency of DNA copying is important during the reproduction. Yes, see, during reproduction, DNA copying should be having consistency. Why they are asking? See, DNA copying is first of all important, that is, it is essential for the formation of addition cellular apparatus. So, when DNA copies separate, each cells get its own cellular apparatus. That means they get their own DNA material. Okay, that is the one thing. The process of DNA copying results in the variation also every time, because see, whatever the DNA material is present in this cell will go for the next cells, but these cells are similar but they are not completely identical. They have some variations. So as a result of this, what happened, the DNA copies generated will be similar, but as I told you, they are not completely identical to the parent cell. They are not completely identical to the pre-existing cell. Okay, so this concept based on DNA, they can ask any question. So in this year paper, this question is little bit tricky. Okay, so the question which I am discussing with you right now is little bit tricky. So this is from the lesson life process. How phototropism, thigmotropism, chemotropism are coordinated the apparent movement of creepers. that is nothing but climbing of plants towards the particular direction they are asking. See, creepers, these creepers, they are the plants which climb up the other plant, or fences by the means of tendrils. Okay, tendrils are the soft green parts of the plant, and when they come in contact with any support, the part of tendril which is in contact with the object does not grow rapidly as the part of the tendril, but the part of tendril which is away from the object will grow rapidly. Why? Because the tendril, they need some time because they circle. Okay, so this causes the tendril to circle around the object. If there is an object here, what happens? So the plant goes like this, they circle like this. Okay, so they go around the object, and thus what happens? They cling to it. So these plants respond to stimuli slowly by growing into a particular direction. So that is why it is phototropic. Okay, and because this growth is also directional, and it appears as if the plant is moving. Okay, this is what you have to write about this, children. See, actually the answer is already given in the textbook, but so they have little bit modified the question. Here you can see that the plants is, that means the tendril is moving like this based on the light which is coming towards the plant. Okay, so this is a directional movement, and as light is passing, it is phototropic movement. Thigmotropic, because why? Because tendril is moving with respect to the object here. Okay, so all these are the reasons for this answer. Okay, yes. So moving to next 36th question, they have asked draw a neat labeled diagram to represent the human brain. They have asked you to label the cerebrum and cerebellum. So you want to label this part and also this part. So this diagram is asked for 3 marks. Okay, so moving to 37th question. 37th question is the 4 mark question. Very simple question, and the answers are also very simple here. The question is, what is the important function of villi? Where are villi? First you have to know, and important function of alveoli in the body they are asking. See, first of all, villi, they are the finger like projections, and they are present in the small intestine. So it helps to increase the surface area of the small intestine, and these villi help basically to absorb the digested food in the small intestine, because we say that small intestine is the site of complete digestion. Okay, small intestine is the site of complete digestion, so it helps in absorbing the digested food. But when I talk about alveoli, they are present in the lungs. They are the tiny balloon shaped air sacs. If you look at the lungs, see, these are the bronchus and these are the bronchioles. So at the end of bronchioles we can see the alveoli. So these are completely covered by the capillaries. Okay, so what is the major role of alveoli? Alveoli are surrounded by capillaries. They help in exchanging the carbon dioxide and oxygen. What do they do? See, these capillaries contains the carbon dioxide. These carbon dioxide will be thrown out, okay, to the bronchioles, okay, through air sacs. It will be sent out through bronchioles and then to the bronchus, and oxygen which is brought by the bronchus to bronchioles will be entered into the alveoli. So this is the first main surface medium for the exchange of oxygen and the carbon dioxide in the lungs. Through alveoli only the gaseous exchanges takes place. So this has been asked, and next they have asked, write the explain the function of the nephron they are asking. Okay, so nephron is actually, it is the basic filtration of the kidney. All the blood which is present in your body, okay, which has to get filtered, will enter to kidney, and the nephron which is present in the kidney is going to filter it. So what do nephron consist of? They consist of a tubule which is connecting to a collecting duct. So one end of this is connected to a cup shaped structure called Bowman's capsule. So let me write the rough sketch of it. So this is the Bowman's capsule. So inside the Bowman's capsule there is glomerulus, which are the thin capillaries. So these glomerulus and Bowman's capsule has connected to a PCT, we call it as proximal convoluted tubule. Okay, so now what happened, this PCT is further, it looks like this. Okay, these are completely surrounded by the blood capillaries, and finally this leads to a connection called as collecting duct. I just wrote a rough sketch of it so that you will get an idea. Okay, so every Bowman's capsule, this region has a cluster of capillaries. So within this cup shaped structure, So blood which enters to this region, that is to the glomerulus, okay, it start filter in this region, okay. So where does it enter? It enter through the afferent arteriole, through renal artery it enter, and this comes out in this region. So this type of connection will be there, okay. So I cannot write the complete diagram now because as I am just discussing the paper, you can refer my previous video to write the neat labeled diagram of nephron, okay. So you must know what are the parts mentioned in the nephron. So now when I talk about the functions of nephron, okay, so it undergo majorly three functions. One is the filtration, the second one is the reabsorption, and the third function is we are talking about the secretion. So when I talk about the filtration, see the blood get filtered in this region, okay. Where do the blood get filtered? The blood get filtered in the Bowman's capsule, okay. So through the Bowman's capsule the blood enter to that is glomerulus, and the blood filter in this region. So once the filtration is taking place, what happened? The materials enter to PCT. So in the PCT what happens, you know, the needed materials, like the material which is important for our body get reabsorbed. Which are those materials? The glucose, amino acid, salts, when it enter to PCT, what happened? So they get reabsorbed. So this process is called reabsorption. So the unwanted materials finally enter into collecting duct. So what are the unwanted materials here? We call it as nitrogenous materials. These nitrogenous materials will enter into collecting duct where the collection of urine takes place, and every collecting duct is finally connected to the hilum, we call it as hilum, that has a connection to the further the structure, that is in the kidney we have ureters, urinary bladder and urethra, okay. So these every collecting duct has a connection to ureters. So through ureters what happen? Urine will flow out. I just said the rough idea, my dear students. I think you can understand very clearly, okay. So in my previous videos when you look at the definitions, the complete explanation is given about the nephron. So as I am discussing the question paper, I am just giving roughly the idea about what is the function of nephron. So the other question they have given here, if you don't want to attend this question, you can also attend what is the importance of transpiration in plants, or how does translocation of materials takes place by the phloem tissue, okay. Yes. So when we talk about the importance of transpiration, see it helps in absorbing, okay, and also it makes the upward movement of water. It helps in the absorption of water from the roots, and also it helps in the upward movement of water and minerals through the xylem vessels. We have xylem vessels and xylem tracheids, okay. These two will help in the absorption of water and also the upward movement of water. This transpiration also help in the temperature regulation. These are the very important 2 points that you have to write when they talk about importance of transpiration in plants. So moving to next, how does translocation of materials takes place in phloem tissue? See, for translocation, first of all, what is translocation? Movement of the transportation of food materials from leaves to different parts of the plant body, we will call it as translocation, okay. See, food molecules which enter through the phloem through the CU tubes, okay. So they get transported to all the parts of plant, including the roots. They get transported. One more important point I would like to add at this moment: translocation. When the food is getting transported in the plant, they transport in the bidirection, okay. So the translocation is mainly achieved by utilizing the energy in the form of ATP molecules, that is adenosine triphosphate, okay. So this provides osmotic pressure, okay, and due to this osmotic pressure, what happens? The required food will move both the upward direction as well as the downward direction, okay. So mainly you have to know translocation is the movement of food molecules in the phloem. This will be achieved by CU tubes present in the phloem. The true translocation, the movement of food takes place. So this will be achieved by using the ATP molecules, and when this is happening, what happens? The movement of food takes place in both the direction, that is through upward as well as the downward direction. And the last question which they asked for 5 marks is, see, very easily they have asked from the hereditary and evolution for 5 marks. The question is tall pea plant, that is when I say tall pea plant, you have to write the gene of it, which is represented by capital T and capital T, and dwarf pea plant, which is represented by small t and small t. So what type of plants will be obtained in the F1 generation, they are asking. See, when we cross between a dwarf pea plant and a tall pea plant, so we get something like this, that is capital T and small t. So the plant which we see in the F1 generation are tall pea plants. All the plants which we see in the F1 generation are tall pea plants. Because why? We can see there is dominant trait present here. So dominant trait, what does it do? It will dominate over the recessive trait. Okay, wherever there is capital letter, always that trait will be expressed because it is a dominant trait. So in F1 generation, we will see all tall pea plants. Further, they have asked to write the genetic makeup of this progeny. Yes, see, this is what you are going to write to represent the F1 generation, and further write the F2 generation of it. Okay, so that is the genetic makeup. So, and when we write this, you have to know what is the genotypic ratio and the phenotypic ratio of it. The genotypic ratio of F1 generation, F2 generation of monohybrid checkerboard is 1:2:1. Why? Because see, we can see completely homozygous tall pea plant 1 set, and heterozygous tall pea plant 2 set, and completely homozygous dwarf pea plant 1 set. So it is 1:2:1. Okay. When I say phenotypic ratio, how many total tall pea plants are there? 3 tall pea plants. And how many total dwarf pea plants are? Dwarf pea plants are there? 1 plant. So that is the phenotypic and genotypic ratio. Always you have to remember, either they may ask in 1 mark, 2 mark, 3 mark, or this time they have asked for 5 marks. So other question which has been attached to this, the final question is, forelimbs of frogs and the wings of birds, wings of bat, forelimbs of lizards, pair them according to their analogous and homologous organ. First of all, you must know what is homologous organ and analogous organ, children. See, when I say homologous organ, they have the similar origin. Their body designs are similar. Okay, their body designs are similar, but they perform different function. So body designs, similar body designs are there for the homologous organs, but they have different functions. They perform different function. Okay, see, when I look at the frogs of, that is forelimbs of frog, they use the forelimbs for hopping purpose. But when I talk about the forelimbs of lizard, they use for crawling purpose. Okay, so but their basic body design remains same, but their functions are different. When I talk about analogous organ, see, analogous organ here, they have different body design. See, when I talk about here the wings of birds and wings of bat, the body design, when we look at the internal structure of the wing of a bird and wing of a bat, their body designs are different. Okay, they are having the different body design, but ultimately when we look at here, see, the wing of bird and wing of bird, they both serve for same purpose, that is, wings of both are used for flight purpose only. Okay, so that is the reason they are called as analogous organ. Understand? Homologous organs have similar body design, they perform different functions. Analogous organs have different body designs, but they perform the same function. So this is all about the biology answers. So I am telling you once again, it is very important to go through all the lessons, because already I have told you, I have given an idea about the blueprint of the the model paper 2023. So those who have joined the live just now, once again you can note it down, the blueprint of all the lessons. So it is very important, my dear students, to know the blueprint of all the lessons. See, for chemical reactions and equations lesson, note down, once again I am repeating, for chemical reactions and equation, I will write it in short form. For chemical reactions and equation lesson, they have allotted 7 marks. Okay, for acids, bases and salts lesson, they have allotted 2 marks. For metals and non-metals lesson, so they have allotted 5 marks. And for carbon and its compounds lesson, they have allotted 6 marks. For periodic classification of elements, they have allotted 4 marks. So totally for chemistry it is 25 marks. Totally for chemistry it is 25 marks. So when I talk about the biology, see, biology life process, for life process they have given 6 marks. 7 mark questions. 7 mark questions are being given in the model paper from the lesson life process. So moving to control and coordination lesson, from this lesson they have given only for 3 marks, that is the human brain diagram. Next, moving to how do organisms reproduce. So how do organisms reproduce, from this lesson they have given 7 marks question. Heredity and evolution lesson, so heredity and evolution lesson, from this lesson they have given totally 5 marks, and our environment lesson they have given for 2 marks, sustainable our environment they have given for 2 marks, sustainable management of natural resources they have given for 3 marks, totally biology 27 marks. So this will help you to decide that how and which lesson I have to complete for the first. Okay, yes. So when I talk about the physics, let me talk about the physics now. So physics electricity lesson, okay. So physics electricity lesson. So for electricity lesson or from electricity lesson, they have given 7 marks questions, okay. Then from magnetic effects of electric current, they have given 6 mark questions. From light reflection and refraction from this lesson, they have given 7 marks question. Human eye and the colorful world, from this lesson they have given 5 mark question. Sources of energy, they have given 3 marks. So totally for physics they have given 28. So 25 plus 27 plus 28. So science totally it is for 80 marks. And question paper pattern, as you have seen, you will get the same question paper pattern. Physics questions are they are being separated from chemistry and separated from biology. Okay, I hope you found this entire session useful, my dear students. Yes, okay. So in my next video, I will be discussing more about the important concepts which will be asked in the final examination from every particular topic. Okay. So if you are finding the questions are not clear, please adjust your video. Okay. So in your YouTube, if you click on the settings button, you have an video quality, keep it for high resolution, definitely you can watch it. So that's all for today's video. I'll be meeting you in my further video, so discussing the more important questions from every single lesson. Thank you so much for watching my video, my dear students.